And our next speaker is Michael West, and he's going to give a great talk. Michael has an amazing background. In fact, his background is in stem cell research, and basically it's interesting that the work that he was doing spurred this organization called CIRM that raised over $5 billion, and that's with a B like Bob, $5 billion for stem cell research here in California. And Melissa King was on the vanguard of that effort, and which is successful. Melissa, could you stand up, please? Can you please stand up? Yeah, thank you, Melissa. Amazing job. Thank you so much. And yeah, and we're gonna. What's that? Hold on. Okay. Oh, we're flickering here. Okay. Oh, you know what it is.
We're supposed to do that, right?
Yeah, well, I'm going to give you this, and let me try to figure this out. Okay, it's our first technical issue of the day. Here we go. Hold on to that. All right, let's start this. Can you get out of the full screen mode, please?
Sure.
Very vibrant.
Very vibrant, yeah.
So, hi. Hello, everyone. Oh, you're going to try a different kind of an output?
Yeah.
Well, we'll see.
Okay,
There you go.
No, Paul Spiegel pointed out, what was it, Paul, about AI and aging? What did he say?
AI is easy, AV is hard.
AI is easy, AV is the really tough one, okay? Thank you, Paul. So true.
Yeah, okay. Don't do this? Why not? Oh, okay, I see why. All right, well, I'm going to talk about this provocative topic of cell immortality and age reprogramming and all that kind of stuff. As Steve mentioned, my background's in stem cell research, but also embryonic stem cell research in particular, but before that, telomere work. And so I'm going to walk you through some of that. Of course, telomere is relating to the cell immortality thing. I've always thought about the problem of aging. I started working on aging in 1980. Went off and did a PhD in the Hayflick phenomenon, studying the Hayflick phenomenon. And I wanted to understand the fundamental molecular mechanisms behind aging, as some of the speakers have alluded to. One of the first things that struck me, when you think about, of course, we're made of cells, right? I'm not trying to state the obvious, but so cell biologists, we just think about cells, and it became quite attractive for me to think about the dichotomy between the reproductive lineage of cells and the cells that make up you and me. So the cells that make up our body are called somatic cells, comes from the Greek word soma, of course, which means body. And this is how I thought of it. So if we look at this picture, this is a daughter and her mother and her mother's mother and her mother's mother's mother and so on. But of course that lineage goes back to her, the mother's 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 for millions of years. And there's a lineage of cells, if you connected the dots, that go back to the origin of life on Earth, the common ancestor to all of life on Earth. And so first when I started thinking about that dichotomy, I thought, well, wait a minute. All these theories about aging when I entered the field in 1980, which would have been talked about, were there seminars here in 1980? I don't know. Yeah, there were, right? Yeah, right. Greg remembers those days. Greg Faye, who's sitting up here, we were talking about this stuff, I remember way back then, and neither of us believed the prevailing theory at the time of free radical damage, right, Greg? We used to talk about this. We just don't buy it. And the idea of entropy and wear and tear, and part of the reason I was agreeing with Greg on that is life has this, not just appearance of immortality and the constant succession of similar individuals in the soma you see here, but literal immortality through the germline. So if you say it's free radical damage and wear and tear that inevitably tears life apart through entropy, then why does this continue? It just didn't make sense to me. So So let's understand the dichotomy. So the first thing most people think when they look at aging, they think it's too complicated, we'll never understand it. But sometimes there's a surprise. What we think we see is a reflection or an image generated by something that we didn't expect. And so let's just talk about this for a minute. So what we're saying is life began on Earth with single-celled organisms that were essentially immortal regenerators. Tetrahymena, the little ciliated animal that you see in pond water animal, can reproduce through binary fission indefinitely. So it's in that sense an immortal regenerator. And then the thought was we evolved into multicellular animals like these volvox here. You can see where they've now bifurcated into germline soma. So some cells are destined to carry the germline cells as sort of a transport vehicle and protect them perhaps from sunlight or help them move through the water or whatever. But that was the first. There was a German scientist, August Weismann, who said that was the origin of aging and death in the soma. That first step where some cells helped other cells perpetuate themselves indefinitely, right? It get a little more complicated, like these little flatworms. I used to collect these as a kid. I love these animals. Here we did a time lapse. These is real images with a little bit of help from computers. Watch the top. Watch the eyes appear. Isn't that amazing? See, isn't that cool? I love these animals. So they're immortal under the edge of the knife, we say. You can cut them in half, cut them in half, cut them in half, cut them in half, and they just keep going and going and going. So even in the soma here in these animals, they're immortal regenerators. They have telomerase, I'm ahead of myself with telomerase, but the cells don't age and they can regenerate. All right, so what we think has happened in the evolution of life on Earth is some of these primitive animals maintained that immortal regeneration that was present at the dawn of life with single cells, and then we started restricting the immortal regenerative potential in the soma for reasons I'll get to in a minute. And so humans have immortal regenerative potential in our germline, and we have some regenerative potential early in our development, but the immortality part is turned off very early on. Why? My belief, and I think a lot of gerontologists believe, that the why is not because of the misbehavior of two humans 6,000 years ago, but is rather just a tumor suppressive mechanism. So nature selected for repressing immortal regenerative potential in the soma to help prevent cancer. August Weismann, the scientist in the 1800s, said it was just for lack of use. We didn't need it in the soma. Now we think it's antagonistic pleiotropy. So nature selected for a potent tumor suppressive mechanism, and that's turning off both cellular immortality and regenerative potential. So the question is, how do we learn the lessons and transfer that technology? You have a technology transfer department at UC Berkeley, right? Well, how do we do a technology transfer from that and learn lessons from the immortality of the germline and its immortal regenerative potential? You notice I call it the pharmacon athanasiou. It's a very ancient term. We won't go into that today. We don't have time. Very early on in ancient Egypt and ancient cultures, the mystery religions, they knew about this dichotomy, and they turned it into religious beliefs. They called it the drug of immortality that they were seeking. All right, so the first idea is, can we capture the cells? So in the mid-90s, I tried to grow some of these cells from developing gonads of humans. I couldn't get them to grow. I couldn't get them early enough, and I formed a collaboration with a couple groups, Jamie Thompson in Wisconsin and Roger Peterson here in San Francisco. And the result was the first isolation of human embryonic stem cells. These cells are the germline. They're immortal, and they're pluripotent, as we say, or they can become all the cells in the body, so they can branch out in the dish, into everything. And what I saw in that was, okay, that's a simple concept. If you're, Aubrey showed the old car that we can keep going by replacement. Let's do cell replacement therapy. So if you need, what are we looking at here? Bone, or what? Bone's easy to make. If we need, we need neurons for Parkinson's, these cells can make them. And there'll be young cells, and because the pluripotent stem cell still retains that immortal potential of the germline, we can propagate these pluripotent cells forever and make young cells forever of any kind, genetically modified in any way. So this became a platform called regenerative medicine, and I'm calling it here regenerative medicine 1.0 because we're now on to 3.0 or something. Well, what could we do with this? Well, one of my favorite ideas, I like, there's a cell type in our body called brown adipocytes that when we lose them, they throw our metabolism off balance. So I could eat ice cream all day long when I was young, and what happens? And now it's, right, diabetes, type 2 diabetes. We know this is the loss of brown fat cells, and we can make those cells. We can make beating heart muscle. People love to see these videos of, these are not mine, but I've got a video of my cells taken back in time and turned into, baby Mike West beating heart muscle. It's an amazing thing. So that's one idea, how we can learn from the germline. Another is, what about this immortality thing? So I started out my career studying cell senescence, as some of the speakers have spoken about. So here's young and senescent cells, the Hayflick phenomenon, as we call it. And so the idea was the immortal regenerative germline is shown in red. And then, remember I said somatic restriction. Somatic cells, the first step here, I'm going temporally, in development, about 2 weeks after the egg is fertilized by sperm, telomerase shuts off. At least functionally, if not transcriptionally in most cells, and the clock starts ticking. So now we've got a clock ticking, and you guys probably have heard about the telomere. So my first company, Geron, we put all of our chips on the concept that the telomere was the clock of cellular aging, or the Hayflick phenomenon. And the idea was there must be a gene out there, or genes. We didn't know how many. That would have been a bummer if it was like DNA polymerase with 23 components. Yeah. But the kinetics, cells can immortalize in cancer, and the kinetics looked like a single hit for immortalization, so we thought there's probably one gene. That was the gamble. And we eventually found it. This was before DNA was sequenced, so it was a very $30 million project to find this gene. And the catalytic component, as this little video shows, makes the telomeric repeat, which is rewinding the clock of cell aging, and telomerase immortalizes cells, so it's an immortalizing enzyme. So that worked. So what about developmental aging? Well, along came nuclear transfer. In 1997, Dolly the sheep was cloned, and I want to understand this. So what is cloning? You just saw a little video clip. Maybe I'll show it again if this will back up. Here we take an egg cell, and we stain it with Herx dye and put it under ultraviolet light, and watch, we go in, that little glowing object there is the chromosomes. We removed the DNA from the egg cell, so now it's just cytoplasm with no genetic instructions. If you add then a floating fibroblast, they call it nuclear transfer, but this is a whole cell, actually, a fibroblast in suspension. And if we then add that DNA, or the cell, in this case, into the zona there, and give it a little push so the membranes are in contact. And then we can electrofuse it. We give it a little shock. Robin Cook wrote a novel called Shock, and that was referring to cloning, and it was that little electric shock we give them. And then something in the egg cell acts like a time machine. And a fibroblast, or a cow, moos, whatever. And I so looked at this and I thought, holy cow, that's... what? I mean, you know, scientists didn't believe this at first. When Keith Campbell was the genius behind cloning, and he thought this would work, but no one in the world thought this would work. And mammals, what? Differentiation is completely reversible. Developmental aging, that is completely reversible. I mean, amazing. Well, then with years later, you know, this became, what's in that egg cell, that acts like a time machine? As you guys know, it's inevitable that we'd figure out what those genes were that did the reversal of the clock of developmental aging. And we have now iPS cells where you can take a fibroblast and add some of these dominant pioneering transcription factors, and they do what the egg cell does, right? Take cells back to the beginning of life. We published that it reversed developmental aging here in 2010, for instance. And now you probably have heard that there are these immense startups to apply this technology directly to a human being. Now, we don't want to, 5 minutes, don't do this to me. We don't want to take cells all the way back to like sperm and eggs. Yeah, that wouldn't be cool in a human being. So the idea is partial reprogramming, as it's commonly called. And I'm going to zip here. The problem is, now here I'm going to challenge this field a bit. The problem is partial reprogramming, blasting a human tissue or a human being with these factors that can take cells back in time and reverse developmental aging. They don't do two things, and that's a fundamental flaw. One, they don't reactivate telomerase. So telomerase would only be turned back on to reset your telomere clock if you went all the way back to pluripotency. We know that, at least in humans. Mice might be different. The second problem, and this is to me a bigger problem, is off-target effects. Although these are pioneering transcription factors and you can make iPS cells, in a tissue, you know, like 1% of the cells would be effectively reprogrammed, and you're going to get all these off-target effects. We need to have a far more precise understanding of what we're trying to reverse, because the intent is not to make iPS cells. It's just to take cells back in time. So we undertook a big project at AGEX to study what is early in development that is our target. How far do we want to take cells back? If you go back when the body's first forming, cells will regenerate. And we thought that's what we want as the target. We want immortal regeneration. Remember I started out talking about, so we want telomerase, which we'll have to do independently, but we want to induce regeneration so the body can repair itself. And there's a clock called the heterochrony clock. It was discovered by Ruvkin, and it's these genes like Lin28, LET7, and they control the developmental timing in animals all the way through C. elegans to mice to humans, as you can see here. And we believe that animals like the Mexican salamander, the axolotls, that can profoundly regenerate limbs and brain and everything else, are stuck early in that developmental clock and not progressing to adulthood. And by targeting that clock, we can reset these two clocks of aging to restore immortal or extended life regeneration in humans, and we think that combined that will have a very profound effect on chronic degenerative disease. So 80% of healthcare costs in the United States are chronic diseases, and aging is full of them. We've had several speakers speak about this problem because they're chronic. The healthcare costs are just, 80% of the several $trillion healthcare costs in the United States. So if we could induce a human to do what the salamander can do, the salamander I showed you can do, by unlocking that regenerative potential, by taking cells back in time, by a precise regulation of the hub genes that control the regulation of this regenerative potential, we think we could do something rather remarkable. And my little startup, LifeCraft, we're taking these heterochrony genes, and it's a little, I don't want to get into all the science, it's a little complicated, but there's a break gene, RNA actually, a microRNA, the first discovered microRNA, LET7, that is like yin and yang. It turns off these regenerative genes here I'm showing you by binding to the 3' UTR and making them be destroyed. So it's like a switch that it's hard to switch back. It's a one-way switch. So when the animal develops into adulthood, which the Mexican salamander never does, but as we do typically, the LET7 switch is dominant and turns off regenerative potential, but it can be overridden by the re-expression of these embryonic genes. They are triggered by partial reprogramming, but in a very off-target way. So it's doing on-target reprogramming, and one of the special tricks we employ is to eliminate the binding site of LET7 so it cannot stop these genes from working. What I've described is there's a lot of people involved, and I want to give credit to this. Jaron, the telomerase team, Cal Harley, who's now gone, we lost him. Woody Wright, we lost. Bill Andrews is still around, last I knew. Tom Check's team managed to find telomerase in euploides, and that was quite helpful for us finally finding the gene in human. Of course, the embryonic stem cell thing, you probably heard of Jamie Thompson and Roger and so on. At Advanced Cell, where we did the nuclear transfer work, here's some of my collaborators and so on. Many people that helped with this. I just want to go step back again and talk a little bit about how amazing life is. This Science and Immortality was a lecture given by the famous physician William Osler in 1904, and he said about the germline, and I'm showing you colonies of embryonic stem cells. This marvelous embryonic substance is eternally young, eternally productive, eternally forming eternally, by the way. I mean, maybe not forever in the future, but so far, forming new individuals to grow up and perish while it remains in the progeny, always youthful, always increasing, always the same. Thousands upon thousands of generations have arisen in the course of the ages were its products, that's you and me, but it lives on in the youngest generations with the power of giving origin to coming millions. The individual organism is transient, but its embryonic substance, which produces the mortal tissues, preserves itself imperishable, everlasting, and constant. It's amazing. I like this quote, too, from Richard Goss in a book on regeneration. If there were no regeneration, there'd be no life. We wouldn't be here. But if everything regenerated, there'd be no death. Something to think about. Thank you very much.
Thank you so much, Michael. There you go. That was amazing. And just Greg Cole wanted to be a physicist in the beginning, and the physicists have something called a beautiful equation, right? There's something of beauty in physics. And I think there's definitely something beauty in biology as well. And I think a lot of us should read some more poetry, because that's how we can convey, or that's how we can communicate with the public, okay? Because if you can say something poetically, I think it really sticks. And obviously all the science and so on and so forth, it's difficult to explain that to some layperson. But poetry, I think most people understand. So I just want to put that out there. All right, questions. Okay, gentleman back there. There you go. Okay, speaking to the little nub over there.
Sure. Hi, I'm Byron. I asked a question about germline yesterday because I'm very fascinated with it over time, especially like the nesting doll of like mothers inside of grandmothers while inside the baby. So like your cells starts really early, like who you turn out to be chronologically starts much earlier than we thought. But basically, I've been working on a balance timer at death versus life. Like, what are these signals that are regenerative or opposite? And so all these clocks combine, do they surmise into a single overarching universal equation for aging?
Okay, hold on a sec, Mike. We're going to get you back here because, again, there were people on the stream here. Hang on. Okay, yeah. People are on the stream, so they need to hear what you're saying, and you need the mic for that. I'll hold on. Okay.
It is, again, my hearing is bad. So as I heard you ask about, is there a universal arching theory of aging that's emerging from this? Is that what you're saying?
Balance timer between aging and death components in the body, either like regenerative or degradation. Can we use that universal, all these multiple clocks throughout the body over time and space, can we convert them into a single equation that find the targets over time and space to see where aging occurs? It could be transgenerationally or it could be in a single individual.
So basically, is there like an equation or something like that you could formulate that would tell you where aging begins? And is that about right?
It's like all these signals that we have in our body are either life promoting or death promoting.
Oh, yeah.
Over time, in space and time, from the germline to all the way to 110 years old. And there's like these proteins like TTR, for example, or even an oocyte are either in stasis, and these cells with hate-quick limits very large, like neurons, for example, and like long-lived proteins. Can we find the signals in aging biology and target them directly? Can we start creating a network graph of where to target?
So there's signaling that actually initiate, I guess, regeneration and I guess senescence, and
Yeah.
is there a way to find that clock?
Like one of these timers.
A timer,
like
yeah.
It's like it's a summation of all these different clocks we have. Can we create an actual timer either to like move it forward or backwards in a single person?
So basically change the way the clock is going? Is that what you're asking? Okay.
Reversing and
Right. So basically stop the clock, reverse the clock. Is there any way to do that?
Yeah. We probably need some better terminology. You know, we were talking about clocks and the Horvath clock and all this epigenetic clocks and all this. The clocks I was referring to are those kinds of clocks I think you're trying to describe, which, I mean, the telomere shortening predicts the replicative lifespan of cells. That was one of the early experiments we showed. And it is a clocking mechanism of cellular aging, right? It counts cell divisions. Yeah, well, telomerase will reverse that, then rewind that clock, yeah. Yeah, and in development, what I'm saying in the developmental clock, and so what's so important about the developmental clock is that early on, we can do what the salamander does. Humans do this early in our first 8 weeks of development. If I cut your spinal cord, it'll grow back. Your heart will grow back even later. And that clock can be turned, well, we don't know the clock behind the heterochrony genes. You know, why is Lin28 active all the way up to like 8 or 9 weeks, and then regeneration shuts off in most tissues in the body? Why does it go 8 weeks? We don't know exactly how that works, but we can turn it back on. We can take cells back by re-expressing those heterochrony genes. Yeah, well, yeah. I don't know that there's. Well, let's talk afterwards, because there's. No, I think I understand where you're going. There's really intriguing indications that there's more to the story than what I described here today, and let's talk later. I'll tell you about it.
All right, thank you, Mike. All right, I think we're going to have our panel next. Please give a round of applause for Michael West. Thank you so much. Thank you. And if I can ask your